Okay, we opened up a whole new topic last day, and it's a vast topic. Perception is a word that has many, many meanings, is highly contested, opens up deep philosophical questions, and also is something we really want to understand urgently. We have very many questions in the area. And we noted that for historical reasons, most work in this area has focused on vision. There's far more work done on vision than on touch or smell and so on. We kind of understand ourselves as visual beings in some sense. But vision is not necessarily the best place to ground your intuitions here, to learn how to ask the most basic questions. And so we began by opening up the question of perception, not just looking at vision, but asking, well, when we come to speak of perception, we're asking about the manner in which we're embedded in the world, in which we come to know the world. We're quite familiar with the everyday manner in which we discuss five senses, which provide information. But that framework becomes, when you start to drill down a little bit, it becomes a bit problematic. For one thing, we might ask, well, are we granting the nervous system a distinguished role in the body here? And typically we are. And so we might ask, okay, where do physical events in the world cause something to happen in the nervous system? And that draws our attention to the receptors. And then we find that the story of five senses kind of goes away. It becomes much more complicated. Although we do find receptor surfaces in the ear and on the retina that respond sensitively to meaningful goings on in the world, there's much more to it than that. So we notice that touch opens up into a wide variety of different kinds of information about thermoception, heat, or non-specific pain, or vibration, or stretch, or deep pressure. We noted that taste and smell are not entirely separate. We noted that the vestibular system, which never gets much of a mention and is located in the inner ear, is actually part of the visual system, helping us to understand who's responsible for change in the environment. Is it us or is it the world? We noted that we have receptors in our joints and muscles giving us information about the position and movement of our body. That's proprioception and kinesthesia. And we noted that we have a whole bunch of receptors that give us information about other aspects of our body, how it's functioning, how well it is, are we nauseous or hungry, need to use the loo, those kinds of interoceptive information also belongs here. So it's got much more complicated, right? And then we just had one instructive example, which is more of a thought experiment than anything else. It's a real case study, and this was the box jellyfish larva, which we met, which is like basically a hairy gooseberry that wiggles in the sea. And wiggling just produces random motion, but around the exterior you can see some specific cells, the ocelli, which are photosensitive. And that means they respond to the angle at which light meets the larva. And so the hairs that are associated with these cells adopt a fixed angle with respect to the incoming light, and that generates a distributed rudder that steers the little gooseberry towards the food, because if you go towards the light in its environment, that's where the food is, that's where the photosynthesizing algae are. To call this a visual system might be seen as a bit of a stretch. I think of it as a visual system because it's making use of ambient information that serves its own purposes and helps it get by. But there is no brain here, there's no eye here, there's not even a nervous system here. So when we take this as a place to ground our intuitions, vision starts to look a lot more like touch, in that it's a form of interaction, engagement with the immediate environment in which one is. Now that box jellyfish larva is a very, very simple being, but even the simplest animal, as long as it has any body at all, must be in contact with its world. So the touch is a logical necessity for any embodied being, whereas vision is not. Vision is a specialized form of engagement with the world, It arose largely after an event called the Cambrian explosion, and it requires the development of sensitive tissues like retinas and sensitive organs like eyes. So touch is a useful place to start in thinking about perception because it emphasizes that, well, when we engage with our world, we are in contact with it, and there's different forms of contact. So one way to think of vision is as a form of contact. I find that useful because it helps to avoid a misunderstanding that arises around vision, which is that it somehow has to do with images. And you will hear nonsense peddled in the marketplace about the eyes sending images to the brain, and that is complete and utter tosh. That is fairy tale stuff. The brain is not in the business of images. Images are particular kinds of objects that are very complex, actually. And humans have only been surrounded by images. We're very image literate. We're surrounded by images. But that's a relatively recent state of affairs and is of no relevance whatsoever to animal vision. So we should not think of vision in terms of images. But how should we think of it? Well, an embodied perspective will emphasize the engagement with the world, the similarity to touch, and so on. But there's another and more popular way to approach vision, and that is as a form of information processing. And this belongs in the computational theory of mind. And when we adopt this framework, what we're doing is we're assuming that we can understand our engagement with the world in a manner that separates perception and action. So perception here is a kind of input used primarily to develop representations of the world, and then action is something separate from that, done by the motor system. That is a framework within which one can ask such questions. And it's not an obligatory framework, and it's often presented as if it were the God's own truth. And I want to emphasize in this module that we have available to us in cognitive science many frameworks. And this one is perhaps oversold sometimes. But because most work in visual perception has been done within this framework, that's the one that will guide us as we look to understand the role of the brain here. But we're going to start by looking not at the brain, but at the sun. Because the sun is the source of most of the light, and from a biological point of view, pretty much all the light that's of relevance to organisms. And the sun gives off a vast array of energy, which is distributed over the electromagnetic spectrum. And there's only a small part of that spectrum that we are sensitive to with our eyes, and we call that visible light. But there's nothing in the electromagnetic spectrum that says that's a special kind of radiation. It's just, we recognize it as being light and therefore different from X-rays and gamma rays because of the eyes that we have, because our eyes pick out this particular section. If we had different eyes, we would be sensitive to different ranges of the electromagnetic spectrum. But it's not an accident that our eyes pick out precisely this range, because if you look at this graph, what's shown here on the outer curve is the relative intensity of the sun's radiation over a range of frequencies on the x-axis. And you can see that the sun doesn't give off equal intensity at all frequencies, rather there's a concentration around some range. And shown in the inner curve is the sensitivity of the human eye. And you can see it matches the outer curve extremely well. That's not an accident. If we had a different sun, if we had a different star that had different characteristics, we would have evolved different eyes. So the eye is sensitive the way it is because it evolved in a terrestrial environment in which light comes from a particular kind of star. It's not pre-given. This is eyes evolves. Now there's room for some variation here, and if you look at different visual systems across the animal kingdom, you will find slightly different ranges here. So some animals are sensitive to lower frequencies or higher frequencies than we are, but they all have this general characteristic that they evolved in a terrestrial environment, and so the sensitivities match the environment. We are hooked in. It's very important to realize this. We don't walk around independent of our environment. We are hooked in, and vision is a means of being hooked into our environment. So now let's turn our gaze away from the sun, because it's a bit blinding, and let's have a look at the eye. And the eye, you've probably learned something about this in school. The eye has a determinate form. It has a lens and a pupil, and it lets light in, and that light is projected onto the thin skin at the back, known as the retina. In very superficial respects, the eye resembles a camera. Cameras also have lenses, and cameras work by collecting light which is projected onto a receptor at the back, usually a charge-coupled device. In your digital cameras, it used to be film. And the charge-coupled device or the film acts as a kind of a screen. The retina, however, is not a screen. And the eye is not a camera. And we'll be spending most of our time on what comes looking at ways in which the eye is better understood as one component in a system that is nothing like a camera at all. So the superficial resemblance is misleading in many respects. I say the retina is not a screen. It's true that if I was to look into the back of your eye, I would see a projection there of what's directly in front of you. Except my head would get in the way, it would be awkward. It's also true that if you look into a drop of water, you can see a projection of what's around reflected in the water. That's geometry. But the retina is not homogeneous. It doesn't function as a screen. It has receptors on it that are not evenly distributed, and they are not registering an image. When we take a cross-section through the retina, we find two kinds of receptors there. We find the so-called rods, shown here in blue, and the cones, shown here in red. Now, rods are we've way more rods than cones. And rods are what you find in most of the retina. Rods are completely insensitive to color. So most of your retina is not responding to color at all. Rods are very sensitive to movement, and they work very well in low light conditions. They're very sensitive to contrast. Cones are restricted to a very narrow part of the retina, just a small circle, which is known as the fovea. And that corresponds on the retina to the point at which you are directly looking. And the cones are sensitive to color. And so the cones are responsible for this view we have of the world as colored. And when we look around to check, of course, we're using our fovea to look around at different places. The cones are very densely packed there, and if you were to examine the projection onto the back of the retina, it is most highly focused here. So the fovea is very, very important. It corresponds to the point of our visual attention, where we are looking. Interestingly, there's a gap on the retina where the optic nerve leaves, and you don't have a sense of a gap in your visual field. Once more illustrating that the retina is not a screen and something else is going on. So now we're going to move from looking at the retina and the eye to looking at how the eye is plugged into the brain. The eyes, of course, for humans are at the front of our head, but they don't plug into the front of the brain. The plumbing is a bit weird, and whoever did it, I wouldn't hire them again. The eyes are plugged into the very back of your brain. Makes no sense whatsoever. Something similar happens with the ears. They're not plugged in directly to the nearest part of the brain either. It's much more complicated. So this is a cross-section through a head. We've got the eyes up on the top. And you can see the optic nerve coming down here. And you can see there's a point at which the nerves, some fibers cross and some don't cross. Shown here in blue and red respectively. We'll have a look at that in just a minute. And then there's a couple of way stations before we get to the brain. There's a couple of junction points, what were called synapses. We'll be looking at the brain later on. But there's a complicated route before we ever get to the brain. And when we do get to the brain, it's not to the front of the brain, but to the very back of the brain, here in a part called the occipital lobe. That's where the eyes are plugged in. We might as well get a little bit of brain anatomy out of the way. We're not going to do this in any detail. But down here on the lower right, you can see the outside of the neocortex. That's from an evolutionary point of view, the newest part of the brain. It's the highly folded surface. And it's divided into basically 4 lobes on each side of the brain. The brain has 2 halves. And we're talking here about the occipital lobe. That's this lobe at the very back of the brain. And when we examine the activity of neurons there, they're very deeply involved in whatever's going on in vision. So as we move around, as we look at different things, the activity here changes. The other lobes, we might as well mention them. We've met the frontal lobe before in developmental psychology. We noted that this is a freakishly large part of our body. What the neck is to a giraffe and the bill is to a platypus, the frontal lobe is to a human being. It's also very far removed from our sensory motor engagement with the world, and it's all a bit mysterious. And we'll be looking at the frontal lobe a bit later on. But for now, let's just notice the other two. The temporal lobe is on the side, just behind the ears. We'll be looking a bit at that now. It's also involved in vision. And the parietal lobe sits up on top of the head here. So those four main lobes serve to divide up the neocortex. Hanging off the base of the brain is like a mini brain here called a cerebellum. It's involved in movement, and we won't be concerned with it when we're discussing perception. And there's much more. There are deep structures in the brain, but that'll serve to orient us, and we're looking at the occipital lobe initially here. Up at the top left, we're going to study that business of the crossing of the fibers. And you can see here's a view from top of a head looking at a spoon. And notice the projections here. The green handle of the spoon projects onto the left side of each eyeball. That's shown there in red. And the blue, the business end of the spoon, projects onto the right side of each eyeball. That's a matter of geometry. That's a matter of how light works. Light travels in straight lines. And so the geometry ensures that the right side of the visual field projects onto the left side of each eyeball and vice versa. After the crossing, after this optic chiasma, some fibers cross and some don't, so that all the red stuff is gathered down here and all the blue stuff is gathered up here, with the result that the right side of the visual field projects directly to the left side of the brain, occipital lobe. The left side of the visual field projects to the right side of the brain. I did not say the right eye projects to the left side of the brain and the left eye projects to the right side. That's not what's going on. The visual field is divided. The right side of the visual field projects to the left side of the brain. The left side of the visual field projects to the right side. That's very important because it means that our vision is working in a sensible fashion, orienting us towards the surfaces in our surround, and is not terribly concerned with the implementation details of the eyeballs and the wiring. Now we can follow what happens after the occipital lobe as well. Things do get a bit murky when you start trying to analyze what's going on inside brains. Brains, it should be no surprise, are complicated. But we can track the activity of individual neurons in diverse parts of the brain. About half the brain is affected by what's going on in vision. And when we do so, we find that vision is complicated and there's at least two different things going on. And this is a view known as the dual pathway processing, and it's associated with Mel Goodale and Milner. And you can see here the occipital lobe on the right. You can see one pathway goes up into the parietal lobe over the top. That's called a dorsal stream. And one pathway projects down and around the bottom of the brain, ending in the inferior temporal lobe. That's called the ventral stream. And they're quite distinct. They, as it were, serve different functions, or the activity of neurons in these streams seems to be related to different aspects of vision, because vision is rich. And as we get further away from the eyeballs and the retina, and further away from the occipital lobe, what we find is that the activity of individual neurons has less to do with patterns of light and dark or colour, and more to do with meaning, more to do with abstract notions. So as we get away from this sensory surface, we're seeing more and more meaning crop into the perceptual system. And meaning, it seems to be an important part of the perceptual system. We're always making sense of the world, as it were, not just looking at it, but making sense of it. And that's really important. Now, dorsal and ventral are technical terms taken from anatomy, and the anatomists have a very, very rich vocabulary for describing parts of the body. And I've just put in for your orientation here a helpful dog with a whole list of anatomical terms. You don't have to learn these off, please. It's just there for your reference. But the dorsal, as you can see on this dog, refers to his back, and the ventral to the belly. So the dorsal stream is as it were, coming up over the top of the brain. If the brain had a back, that's where it would be. And the ventral stream is down low, coming around the base and into the sides of the brain. So let's look at those two streams, one after the other. The first one, the dorsal stream, that's the red arrows. As we follow here, what we find is that neurons here are very sensitive to change that's produced by motion. Motion or change on the retina arises from motion generated by myself, as when I walk around, or generated by things in the world, as when someone throws a tomato at me. In the dorsal stream, we find a great sensitivity to this, and that's really, really useful kind of information for avoiding walking into walls and lampposts. This is the kind of information that you need to get around in your environment. And we got a hint of this from the box jellyfish larva. Vision, one of the primary purposes of vision is getting around in your environment, is moving. So this is very, very important. And if you don't like the name dorsal stream, you can call it the where stream, because it keeps track of where things are. It's very important in understanding your position with respect to the surfaces around you. And that guides action, it guides movement. It's obviously a very important part of vision, and that goes on here going up towards the parietal lobe. And it's separate from what's going on in the ventral stream. The ventral stream, which is the yellow arrows here going down and around into the temporal lobe, is sometimes known as the what stream, because as well as not walking into lampposts, we use our vision for recognizing things, don't we? We look at the world and we say, that's a chair, that's my grandmother, that's a spoon. We recognize things, and that's the kind of business going on here in the ventral stream. When we look at individual neurons and their responses throughout the ventral stream, we find them responding to increasingly sophisticated, abstract, even conceptual properties of the world around us, and less to their, again, patterns of light and dark. So it's very interesting to see what's going on here in the inferior temporal cortex. That's shown here up in green. That's the end of the ventral stream. And we find sort of a complex organization of neurons here, which is difficult to figure out. And the way that you figure out what's going on here, this is work usually done on rhesus macaque monkeys, is you insert a very, very fine electrode into individual cells, and then you try to show the monkey various things and try to find out, well, what does the cell seem to respond to? And most cells respond to nothing. But when you're lucky, you find a cell that responds to something, might be something as specific as a banana, for example. We don't only do this with monkeys, we do this sometimes with humans prior to surgery. So if you're going in to do tumor surgery or certain kinds of very invasive surgery necessary for epilepsy, we do this kind of thing just to find out, to orient ourselves, what's going on in the area we're about to go into. And different studies reveal different kinds of sensitivities, and there's something of a lottery going on. It's a bit of a crapshoot. When you initially put an electrode into an individual nerve cell here, you have no idea what you're going to find. So here's some examples that are quite illustrative. Here in the top left, we have a study done from a single neuron in the inferior temporal cortex of a rhesus macaque monkey. And the two lower displays show the activity of this individual neuron, and you can see that it's very inactive except here and here where it's very active. And shown up at the top is what the monkey is looking at. It's looking at the face of another monkey, a conspecific. Now, faces are highly significant to both monkeys and humans. So it's not surprising that we find neurons that are very, very sensitive to faces here. But look how specific this one is. It's sensitive to the presence of a conspecific in profile. It doesn't even respond when the other monkey looks at it straight on. It responds sensitively to the other monkey in profile. That's a very, very specific response, isn't it? It means that this neuron is very finely tuned to something that's very meaningful in the monkey's world, but very finely tuned. Now let's have a look at this display down at the bottom right. Here we've had the good luck to find a neuron which responds selectively to a hand, like this. That's a hand, bing, this neuron fires, and the red stuff is a histogram of the firing. It shows, yay, this neuron is excited by the hand, as it were. Once we find something like this, we can investigate around it. Initially, it's a crapshoot. We don't know what we're looking for. We try hands and bananas and every goddamn thing. But once we find something, then we vary that slightly to try to understand what is this neuron responding to. So the next thing we do is we turn the hand around. The neuron is still firing like crazy. Okay, so it's responding to a hand, and it doesn't matter whether it's the front or the back of the hand. Cool. Now we try it with a cartoon of the hand, a drawing of the hand. It's just the outline, and it's still firing like crazy. So it's recognizing this as a hand, as it were. There's something that's continuous across all these examples. It's not responding to the skin tone or to the hair. It's responding to the shape of the hand. Then we turn the hand 90 degrees. That creates a very, very different visual pattern on the retina, obviously. Different parts of the retina are now being stimulated. But this neuron doesn't care. It's still responding very strongly. That's what I mean when I say these neurons respond to the meaning of something rather than to its visual appearance. So the change brought about by reorienting the hand is of no significance, and so the neuron doesn't even respond to it. But now we move the hand away a bit, and the response of the neuron goes down. So it's orientation insensitive, but it's size sensitive. That's very interesting. And we replace the hand by an outline of a mitten, and the response pretty much goes away. So the fingers are an important part of this. Hang on, maybe it's just responding to fingers. Maybe we've misunderstood things. So we try a variety of finger-like things down here, and the neuron doesn't respond to them at all. None of these are hands, of course, they're just finger-like. And we try other things like faces and shapes, and the neuron doesn't respond. So now we've got a lot of information about this neuron, and now I'm going to ask you perhaps an unexpected question. Who owns the hand? It's a bit like, what did the bus driver have in his sandwiches, isn't it? You're all looking at the screen right now. Look at the world around you. Look, there's hands in it. Here's my hands. Your neighbour has some hands. Look around you. Which hands are going to be of a predictable size? In your visual field. Do I have to pass the bong around? Your own hands. Right? Stare at your hands. Your hands are a fixed different distance from your head. Everyone else's hands, they come and they go. Sometimes they're in your face, sometimes they're off in the distance. But if there's one pair of hands in your world that are going to be of a predictable size, it's your own hands. Right? Oh my God, I just found my hands. And this neuron is sensitively responding to a hand at a fixed distance, independently of the orientation. So it's responding to the monkey's own hands. You see, you've got to be a bit of a detective when you're doing this kind of work. And this illustrates nicely that as we go away from further and further from the retina, what we find is that the individual neurons are taking part in something that's much more meaningful than patterns of light and dark. They're responding to more and more abstract properties. And this recognition gave rise to the absurd notion that there might be a neuron there that somewhere in your head that responds selectively to your grandmother's face and to nothing else in the world. The so-called grandmother cell. And this was sort of a joke, and I hope it's not true, because if you go out on the batter on a Saturday night and you kill a few brain cells, you might kill that one and then you wouldn't know your grandmother, and that wouldn't be a good thing, right? That would be a bit disastrous. So that's probably not what's going on, but we do find cells that respond to very, very specific things. And there was one case of a human subject prior to surgery for epilepsy, and they were doing this kind of checking, and they found one neuron that responded selectively to pictures of Jennifer Aniston's face. That's the actress from Friends. I have no idea why, but that's how bizarre this gets. Now, faces are, of course, extremely important to both us and to monkeys. And so we find neurons that are very, very sensitive to recognizing faces, and there's a lot more to face recognition than meets the eye. Some of you will have seen this picture over in the science building. You've all seen this. This is Brian Vonson. He's a lecturer over in physics. And this picture is hanging there, and it's hanging upside down to illustrate an illusion known as the Thatcher effect. Because this was first done not with Brian Vonson's face, but with Margaret Thatcher's face. She had no part in the research, I assure you. It's just named after her. Now, you may notice there's something wrong with that face. Right? There's something wrong with that face, and I'm not sure whether you're clear what's wrong with it. But if I turn the face the right way around and I'm doing nothing else, oh my God, it's grotesque. Look at that. It's absolutely horrible. You want to kill it. That's more relaxing. What's happened is, of course, the eyes and the mouth have been inverted. And when you look at it in this orientation, it doesn't look too bad. I mean, now I've told you that you're aware of it, or maybe you knew before, but it's not ghastly, grotesque. You don't want to kill poor Brian there. Now, this is kind of interesting because obviously you're seeing the face here and you're seeing the face here, but there's something different going on. You usually encounter faces right way up. You don't usually encounter upside down faces. And maybe you've spent some time looking at the face of a loved one upside down if you're hanging out on the couch or something like that, or in bed. And faces are really hard to recognize upside down. Your ability to recognize them goes away. So as well as seeing the parts of the face, there's more to recognizing a face that has to do with the integration of those parts into a coherent whole. And then we can use this illusion to demonstrate that that's actually quite hardwired. It's not something that's been done on the fly, and we can illustrate it like this. So if I make this rotate like that, you can see it changes. Its quality changes from grotesque to acceptable, at least not horrible. It's acceptable, acceptable, acceptable. Oh my God, it's grotesque again. And it'll keep doing this, and you will never get, you get bored with it maybe, but it's not going to go away. If we sit here, we're not going to do this, but if we sit here and do this all day, it will not go away. You will not learn anything. So the difference between the interpretation of the face as a whole and the interpretation of the parts is obviously hardwired in there somewhere. So that's the kind of detective work, again, that we have to do as we try to figure out from neurons and from what we know of vision what's going on. So now we're going to get to another piece of detective work, and this is kind of bizarre. It's a bit of a Scooby-Doo story. It's how many of you have ever heard of something called mirror neurons? One. A few years ago, nearly everyone in this class would have heard of them. So they made a lot of noise in the literature, and they've kind of gone away. They were found in the 1990s. They were a chance discovery in a lab in Parma, in Italy. And there's a kind of a dramatic story told about how they were found. You have to understand that these researchers were working with rhesus macaque monkeys again. They're the workhorse for this kind of investigation. And they were working within a specific view of what a brain is, a view that we've come to know as the computational theory of mind. And on the computational theory of mind, the visual system provides information to the brain, which is then used in order to control action. So perception is separate from action. And they were studying individual neurons of the kind that we've just been looking at, but they were studying one in the motor system of this monkey. It was part of the frontal lobe, actually, an area that they knew, they understood to be concerned with movement done by the monkey. And they had followed this, and every time the monkey picked up a peanut, this neuron fired like billy-o, and they knew that. They thought they were secure in their knowledge. Now, lower down the lights, it's late at night one time. The monkey is in the lab, and there's a postdoc coming into the lab late, and someone's left a peanut out in front of the monkey, and the postdoc goes to pick up the peanut, and these neurons are hooked up to a machine that makes a noise, and the postdoc hears the neuron going crazy when the postdoc goes to pick up not the monkey's not doing anything except looking. But the postdoc is going to pick up the peanut, but the neuron is firing as if the monkey was picking up the peanut. And the postdoc gets spooked and goes back and speaks to everyone about this, and they go and do some investigation, and this turns out this happens reliably. So that this neuron now seems to be particularly active when the activity of peanut picking up is going on, but it doesn't matter whether it's the monkey picking up the peanut or whether it's someone else picking up the peanut. Now, if you're working within this framework that sees perception as separate from action, if you're working, in other words, in the very standard computational theory of mind, this is quite mysterious. It seems to provide a direct link between perception and action. And fire up the hype machine, did this get out of hand? So mirror neurons, don't call them monkey see, monkey do neurons, seemed to these researchers to provide a magic link between seeing and doing. Now, let's go back to that box jellyfish larva. There was no link, no separation between perception and action there. And that characterization of it as a visual system comes from an understanding of it as an embodied organism. It's not anything to do with the computational theory of mind. But these guys were working within the computational theory of mind, as most researchers in computational neuroscience do. And for them, this seemed hugely important. To those of us of an embodied stance, I'm going to show my colours here. I belong to the other school. This is precisely what you would expect to find in brains. The idea that there would be a separation is bizarre to me. But if you were working within the computational theory of mind, mirror neurons now suddenly appeared as a missing link, as it were. And so they were suggested that from the observation of individual neurons in monkeys, a hypothetical mirror system was put forward by which we interpret the activity of others. And that might be disturbed, which could give rise to the social symptoms of autism. It might provide a basis for empathy. It might provide a basis for the evolution of tool use. It might provide a basis for the evolution of language. It might provide a basis for the enjoyment of sport. It got out of hand, folks. It really got out of hand. Mirror neurons were suddenly being touted from every street corner as the answer to everything. Now, we've met this kind of hype before. We met it when we were discussing language, and back in the 1980s, people were looking for a gene for language, and so they, on the basis of one experimental finding that turned out to be rather more complex than they thought, there was a rush to publish and declare that we had found the gene for language. And once a story like that gets out there, there's no end to the hype. So mirror neurons gave rise to all this kind of activity. It goes on to this day, but I think people have learned to be more cautious, and the fact that no more than one or two of you have even heard of mirror neurons suggests to me that this hype is dying down. But that's where it comes from. Okay, we're now going to turn to an interesting characteristic of vision, where we're going to link vision to our attentional capacity, our ability to pay attention, or to put it in another way, our ability to make sense of what's going on in front of our eyes. So let's start off with a little video. Here's a French video in which you're asked to follow the coin until the end of the game. Okay, there's the coin, congratulations. And how many of you saw the man in the gorilla suit? There was no man in a gorilla suit. Many of you know what I'm talking about. Many of you will have seen an instructional road safety video in which people are passing basketballs around. There's a white team and a black team, and while they're passing around, and your job is to count the passes, a guy walks in a gorilla suit, does this, middle of the stage, and walks out again. And the point is, a lot of people, when they're occupied in counting the passes, miss the rather obvious man in a gorilla suit. Now, that used to be the case, but it's no longer the case. 10 years ago I used to use that video here and half the class would not see the man in the gorilla suit. So hands up who saw the green pepper. About a third of you. For those of you who don't know what the hell I'm talking about, let's follow this. There. Now watch the bottom left-hand corner. In comes a green pepper. There's a green pepper. And the green pepper is swapped back out again. So there was a green pepper swapped in and out. Many of you didn't see it. So there was no man in a gorilla suit, but a lot of you didn't see the green pepper. The video was called Attentional Blindness, but usually this is called Inattentional Blindness. And of course, every time you do this, some people will see it, but a lot of people will miss it. And the point is that you're engaged in a particular task. You're following the coin because you don't want to mess up on that. And that literally makes you blind to some details that really are kind of important. And this is the reason. This phenomenon of inattentional blindness is the reason that mobile phones are banned from driving, because in driving you're engaged in a very, very important task. And if you pay attention to something completely different, it can make you very blind to what's going on in front of you, literally blind. Irish legislation is out of touch with scientific findings in this regard, because, as I've just described it, I didn't have to mention hands. We're done with hands now, we found our hands. There's nothing in the scientific finding that suggests it matters whether you're touching the phone using your hands or not. What matters is what you're paying attention to. In Ireland, we ended up with legislation that allows hands-free use of your phone, but not hands-on use of your phone, which is not well aligned with scientific findings at all. The scientific finding is that if you're paying attention to one thing, you will tend to be inobservant elsewhere. So, inattentional blindness is one example where we might think, I'm going to suggest the term sense-making is useful now for thinking what's going on in vision. Because you're paying attention to one thing, you're making sense of the scene in front of you in one way, and so you're not making sense of it in another way. Inattentional blindness is one example of how you can miss something that's right in front of you. And here's another one. It's called change blindness. And change blindness is quite different. They share this characteristic that you're missing something that you'd think would be important. But in change blindness, what we find is that if you're viewing a scene directly in front of you, and we're going to use pictures in this case, you can be insensitive to quite large changes in that scene if they coincide with a discontinuity. Now, a discontinuity can be a flash, a mud splash, but it can also be a blink, so that your viewing of the world is riddled with these discontinuities. And if you do this kind of thing and you carefully time the change with someone's blinks, they can really, really miss it. So I can't register your blinks, so I'm going to use a different kind of discontinuity. Put your hand up when you can see the obvious change that's happening from one frame to the next. Nobody. 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 1, 2, 3, 4. There's no man in the gorilla suit, but there's a big change, about 5. Okay, have a look over the Sphinx's shoulder, the big bush that's sort of popping up. Oops. Whoops. Get back. You see this bush here? Now you see it, now you don't. That took a long time, didn't it? So that was rather obvious. We're going to do one more example. Put your hands up again when you can see the rather obvious change going on here. 1, 2, 3, 4, 5. Okay, that's about half the class. If you haven't seen it yet, look at the bar. It's rather obvious that there's big bars going up and down and up and down and up and down. And you didn't. How could it be that you missed that? It's not laziness. It's not stupidity. What's going on? How could you possibly miss something that obvious? Well, we are not machines. We are humans. When you look at a scene like this, you're immediately making sense of it. That's why I say sense making is a useful kind of phrase here. And so your attention is drawn to the things that really matter here, which is the people. So we can record someone's eye tracks as they try to do this task. And here's the registration of the eye track of someone as they try to make sense of this. And you can see where they're looking. You can see they're looking at the faces of the participants, and they're looking at those objects that are of joint attention. We've met that before. They're looking at the table. What's on the table? The bottle of wine. That's very, very important. That's 10 seconds of looking at this, and you can see the eyes going around. Here's the next 10 seconds. And around and around and around and around and around. So this person is looking in one way, and they don't change the way of looking. What you're not doing is scanning like a machine. You're not scanning methodically and mechanically over the entire image. You are immediately interpreting it in meaningful terms, not as a pattern of light and dark, or as shading and colour, but as people sitting down at a meal. Alvin Noe, who is a philosopher who's very, very good on vision, says this. He says, change blindness doesn't show that we fail to experience the rich array of detail that we seem to see. It shows something else. Our ability to sustain perceptual contact with the environment over time is not a matter of there being a picture of the scene in our brains. It's a matter of access, and this is a matter of skill. Look at the terms he's using. Perceptual contact. That reminds me of the box jellyfish larva who's in contact with its environment. And it shows you that when you're using your eyes, you're selectively picking out parts of the world to stay in contact with them. So seeing is clearly not a matter of image analysis by the brain. It's a matter of exploration and engagement with the world. This is a theme we're going to explore more on Thursday, but just in advance, when we turn to phenomena like inattentional blindness or change blindness, they get discussed a lot in the popular press. People like these. There's lots and lots of videos on them on the web. If an explanation is offered for these, and it begins with the words, your brain, walk away. Your brain will, any explanation that starts with your brain will not help you to make sense of these. What's going on here is that you are engaging with the world, making sense of the world, parsing the world in terms that are meaningful to you. You are actively exploring the world. Your vision is a form of activity. And this is something we're going to explore more on Thursday, and we'll get into the wonderful world of visual illusions as well. So we'll pick the story up on Thursday.